Thank you, everybody. My name is Lin Yi. I'm going to present the efficacy and safety study of our brand name nutritional ingredient called NMN. And the chemical name is β-nicotinamide mononucleotide as anti-aging supplementation in healthy middle-aged and older adults. It's a randomized, multi-center, double-blind, placebo-controlled, parallel group, dose-dependent clinical trial. This is for presentation for Aging and the Gerontology Conference 2022. This trial is conducted by, sponsored by ABA Chemical Corporation in Shanghai, China, and also my company, Abinopharm Incorporation, Connecticut, USA. So, in the previous presenter of this conference, Dr. Professor Kennedy, he presented a way to improve the health span and the lifespan, also called longevity or gerontology, by using micronutrients. One of the molecules he presented is NMN. What is NMN? And the chemical structure is present here, which is an endogenous molecule exists pretty much in all mammalian cells, not including humans. Also, many plants has small quantity of NMN, such as edamame, has around 0.5 to 2 milligram per 100 grams, and broccoli has a little less. So a number of other plants have this ingredient. How this NMN act for its function or effect or impact on longevity. Here on the left of the screen shows when we get intake NMN either from a natural source or from a supplementation, we'll convert to NAD in the cells by an enzyme called NMNAT. And there's a number of other precursors in recent years used as supplementation to increase the human cells cellular NAD level. NR is another one, and also the older one is called nicotinamide. That belongs to a series of B2-3 vitamins. So NMN is the most close molecule to NAD. It's one step convert to NAD. So it's a key precursor of NAD cellular biosynthesis. Preclinical study revealed that most of the function of biological function of NMN come from its ability to elevate cellular NAD concentration. So during recent years, through a number of academic labs like Harvard professor David Sinclair and also the University of Washington, St. Louis, Professor Yi's lab, they did lots of work on this molecule and the function of NAD convert NMN convert to NAD. And also after that, around the world, there's a lot of academic research and also commercial industrial company involved in the research on this molecule, the function of NAD, surrounding NAD. There's over 1,000 research papers published so far on NAD, the function of NAD. Most of the preclinical research revealed the NAD has a lot of biological function that could be potentially impact the human disease aging-related disease. This here is published in a review paper in Cell Metabolism in 2019 by David Sinclair. As shown here, there's quite a few all very important disease area was impacted by NAD. And showing on the left side, as you can see, with the people aging, the NAD level dramatically decreased. With the decline of NAD level in human cellular will has direct, it's a core hallmark of aging, as shown on the left side. As a few review paper disclosed, the hallmark of aging pretty much include DNA damaging, irregularity, nutrient sensing, loss of proteostasis, and so on and so on. So pretty much the decline of NAD involved all these hallmark of aging. Showing a question mark is telomere, which previously thought NAD is not involved in the telomere attrition. Actually, in recent research, NAD also involved telomere attrition with the age. So that's the results from preclinical studies, which involve in vitro and in vivo study. But how about in human? Can NMN really help increase human cellular NAD level? Or if it did, how NMN increase human cellular level? So a couple of years ago, published by Professor Brenner, who used to be professor at the Dartmouth College, and recently is in, I believe it's in Iowa State University or University of Iowa. He published a paper in Nature Communication. He thinks, based on his research, he thinks the NMN have to be converted to NR to be taken into the cell and then convert to NAD. NMN cannot directly get into the cell. So that's his conclusion. Later on, in 2019, Professor Yi published a paper, and he found a specific transporter called SLC12A8, which can directly transport NMN into the cell and then convert directly to NAD. Why this is important? Because NMN has to be, since 2016, as a supplement already commercialized on the Japanese market, and later on was introduced into the US market. Now NAD as a supplement is widely available in these two countries and also a number of other countries. So people argue NMN cannot be directly get into the cell, only have to convert to NR. Why we take NMN? Why we didn't take NR directly? This discovery, I think, found NMN can directly get into the cell is pretty important for the industry. So that's a lot of very positive results from the preclinical study. But how about human study? Actually, so far there's only limited results. Here list so far all the published human clinical trial on NMN. The first, everybody agreed, the first one published on human the human clinical trial of NMN is the open label study on 10 healthy Japanese of 40 to 60 years old. This was conducted in Japan by Kyoto University, which was published in 2020 in the Chronic Journal. The material was made by Oriental East Corporation in Japan. And the dosing is 100, 250, 500 milligram oral dosing. This is a single dosing study, so we only dose once and then take a sample, blood sample, and do the test to see the results. So in this study, they didn't report whether after NMN supplementation, the NAD level, blood NAD level is elevated or not. They only reported the metabolites of NMN was significantly increased. They also tested the phenotype after NMN supplementation. They found that there's no improvement in sleep quality or ophthalmic assessment. But they found this dosing, the single dosing is safe up to 500 milligrams. Safe and well tolerated. So this first study is open label study, not a control. So the second study was conducted by Professor Yi at Washington University in St. Louis. And this is a randomized control study with 25 pre-diabetic and obese women of post-menopause. This results published in 2021 in Science. The material also was from Oriental East. The dosing is 25 or 250 milligrams fixed dose oral daily. So they take daily for 10 weeks. And they check whether the impact on humans. In this study, they do find NMN supplementation can increase the PBMC, the peripheral blood mononucleotide cell. And also the NMN supplementation can increase this metabolites of NMN, as this results in similar as previous study. They also checked whether NMN supplementation can increase NAD in muscle, but they didn't find the NAD level increased in muscle after the NMN treatment. They only found the metabolites increased in muscle. They also studied the physical strength. They found the physical strength in muscle is not improved after NMN supplementation, but they found the muscle insulin sensitivity singularly increased. So that's a very positive result for this study. I think the single most important study we found out for this study is the muscle insulin sensitivity and singularly increased. They did a lot of study on that area and the report. Also, they found this study is safe and well tolerated. Okay, the third study is also a randomized control study with 48 healthy amateur Chinese athletes of 27 to 50 years old. This study was conducted by a group of professors at Guangzhou University, Guangzhou Sports University in China, and also published in 2021. The materials came from Jin Harbor Biotech in Hong Kong, China. The dosing is 300, 600, 1200 milligrams, or oral dosing for 6 weeks. The result of the study is not, it's just trying to compare the NMN supplementation plus exercises compared with the exercise alone. What's the effect on acid damage? So the results found out NMN supplementation and exercises plus exercises improves aerobic capacity and the muscle oxygen utilization compared to exercise alone in 600, 1200 milligrams, so the higher doses. but the physical strength assessment showed no difference, except the single leg stance for 600 milligrams. So it's the median dosing looks like is the best result for both studies. So they did a lot of other assessments, only these results show significant change. They also disclosed the dosing up to 1200 milligrams daily, oral daily, is safe and well tolerated. So the fourth one is also a randomized control study with 108 healthy Japanese adults. So this study is also conducted in Japan by University of Tsukuba, published in early this year. The material was manufactured by Mitsubishi Corporation Life Science. The dosing also is 250 milligrams fixed dosage oral daily for 12 weeks. This trial measured sleep quality, fatigue, and physical performance. And the dosing administration is tested in the morning and afternoon. The study only found the afternoon treated NMN treated group will have improvement in lower limb function and shoulderless in older Japanese adults. So they tested a number of other categories, only these two categories found statistically significant improvement. This study also found the material is safe and well tolerated. So the fifth study, clinical study, also published recently by a Chinese company called Abinopharm, Shanghai, China. This study is conducted in India with 66 healthy Indian adults of 40 to 65 years old. Just published in Frontage Aging. And the material was manufactured by Abinopharm, Shanghai, China, with a fixed dose, 300 milligrams fixed dose oral daily for 60 days. This study measured the blood NAD concentration, also the physical performance of NAD supplementation. Unfortunately, the results showing there's no statistically significant in both the NAD level elevation and also the physical function and also overall health assessment. No significant change also in insulin resistance. But the dosing at 300 milligrams for 60 days is safe and well tolerated. The first study is also conducted in Japan on 30 healthy Japanese adults, 20 to 65 years old. This was done by University of Toyama, Japan, also published recently. The material was made by Mitsubishi, again by Mitsubishi Corporation Life Science. It's also a fixed oral dosage of 250 milligrams for 12 weeks. This study was pretty much, in my opinion, the first time in human clinical trial really confirmed NMN can increase human blood NAD concentration, elevation the NAD level. And on top of that, this study also confirmed the blood level of both NMN and the NAD metabolites increased, but they didn't detect NMN concentration after NMN supplementation. This study also confirmed the material is safe and well tolerated at this dosage. The next clinical trial is pretty much published almost at the same time, also the very similar clinical trial with 42 healthy Japanese men. At this time, and only using male volunteer of over 65 years old. This study was conducted by University of Tokyo. Material also, again, was provided by Mitsubishi Life Science. The dosing is the same, and the duration is also the same. The result pretty much is very similar. as the previous one. They confirmed NMN supplementation can increase blood NAD level significantly. On top of that, they also can increase blood level of both NMN and NAD metabolites. At this time, another significant discovery is blood NMN and NR levels significantly increased. So this is also the first time human clinical trial found NMN level concentration itself can be increased. They also did some assessment of physical function after NMN supplementation, and showed some of the customers showed some improvement. Some of them failed to show any improvement. So it's a partial success in this customer. Again, the material is safe and well tolerated. And the eighth recently published clinical trial on human was also a controlled, randomized controlled study. So this study is mostly focused on the pharmacodynamics of on the human. So this study chose 32 obese adults, 55 to 80 years old. This conducted in the USA by Metro International Biotech and published in Journal of Gerontology. This study used a very high dosing strategy, 1,000 and 2,000 NMN. It's a type of very special type of NMN. It's called microcrystalline NMN oral dosing for 14 days. And they also claim this special type of crystalline can help the NMN absorption and bioavailability. The results show indeed at this dosage of this special type of NMN material increase blood NAD significantly and also dose independently. They also detect NMN concentration in the blood, and also its metabolites. This material at this very high dosage is also safe and well tolerated. So this is the last three clinical trial on human. Also, it's very recently published. The top one is open label 16 healthy post-menopause women of 50 to 80 years. This was conducted in Japan by a Finnish clinic. Just published in Glycative Stress Research. This material was also made by a Japanese company called Sciinov Medical Research. The dosing is 300 milligram oral dose for 8 weeks. This study again confirmed NAD level was significantly increased. Unfortunately, it's decreased over baseline. So this is kind of very normal. Also, they didn't detect NAD, the blood NAD. Only detect the metabolites NAD. They've also found some blood biomarkers significantly change over baseline, such as HbA1c, HD, high density cholesterol increased. A number of other biomarkers changed dramatically. Skin condition improves dramatically. And this study was found material is safe and well tolerated. So this is very surprising. Failed to confirm the increase of blood NAD level compared to the previous several studies. Another study also is a controlled randomized study using 31 healthy Japanese adults. So this also conducted in Japan, this time by Mitsubishi Life Science itself. Their purpose is trying to confirm the safety at high dosage. This time they use 1,250 milligram oral dose for 4 weeks. So this purpose of this study is for safety. They confirm the safety of the study. There's no adverse effect. Also, toxicity. Ames test is negative. All the blood marker is within normal range. So it's confirmed safe and well tolerated. Lastly, there's an open study using, also conducted in Japan, using 10 healthy Japanese adults. But this time they use a 300 milligram single dosage by injection, intravenous injection. So this is the first time in a injection to see what happens. They found their results is very interesting. First of all, they confirmed blood energy level significant increase. That's inconsistent with a few previous studies in Japan. And also they found no abnormal in vital sign in the clinical parameter except triglyceride was favorably decreased significantly. So this is a very good study on supplementation by injection. Other than that, all the biomarkers shows the material by injection, the high dose injection, 300 milligram single injection is safe and well tolerated. So far there's more than 10, around 11 human clinical trials published. I would say the result is a mixed result. The major primary conclusion from all the study is the material NMN is safe and well tolerated up to almost 2,000 milligrams oral dose daily. And one study injection 300 milligrams is also safe and well tolerated. And several of the studies confirm NMN at fixed dosage can indeed increase the blood energy levels. The number of other studies didn't confirm any blood energy level did increase or not. Thirdly, the number of studies trying to measure the physical impact by NMN supplementation, but mixed results. Some of them showing some of them is successful, some of them is failed to have any physical improvement, and some of the results is contradictory from one clinical trial to the other. So we, by the time we design our study, there is only one clinical trial published. That's the one. And the first one didn't show any blood energy concentration increase. So our designs is based on those previous clinical trial, and we did design the randomized multi-center double-blind placebo-controlled and parallel dose independent. We used 8 healthy adults of 40 to 65 years old, both men and women. This study was conducted in India, so all the volunteer is Indian nationality. 60 days in the trial is 60 day duration. Dosing is using 300, 600, and 900 oral daily intake of our material, which is a fine crystal powder manufactured by ABA Chemical Corporation in Shanghai, China. So the trial site is Notice Healthcare and Aesthetic Clinic, and also Standard Asia, both in Pune, India. Strictly follow the principle of Declaration of Helsinki, ICH, GCP, and Indian New Drug and the clinical trial. The trial was managed and monitored by ProRelics Service LLP, which is a clinic research organization in Pune, India, and Abinopharm, Connecticut, US, which is my company. The trial was registered with clinicaltrial.gov registration number shown here. Also, clinical trial registered in India. ABA Chemical Corporation in Shanghai, China, fully funded this trial. So that's financial funding and also conflict of interest is shown here. Primary goal of the trial was to evaluate the blood NAD level of supplementation with those independent regimes. The secondary objective was to assess the safety and the tolerability. The trial was also designed to assess clinical efficacy of physical functions by using 6-minute walking test, biological aids, insulin research by using HOMA-IR, and also general health by using SF36 questionnaire. Here is our trial flow chart. Basically, the content of this flow chart pretty much discussed in previous slides. We're using 8 healthy volunteers. So they divided into 4 groups. Each group have 20 volunteers. Placebo, 300 milligrams, 600 milligrams, 900 milligrams. So our investigational product is capsules. We compartmentalize in both placebo and the NMN powder into the same capsule, same type of capsule. There's no distinguish between the placebo or NMN. And then volunteer took the product by instruction. And randomized, this material was randomized by the CRO company, randomized and double blinded by this CRO company. So these are our results. Our first primary results, which is the blood NAD concentration. After first make clear that we made the total concentration of NAD plus NADH as our blood NAD concentration by using a commercial NAD test kit. It's a validated test method, which is a colorimetric analysis of blood sample. The statistic significance is set at p-value less than 5%. Using mixed model of repeat measurements. So this graph shows this is the intergroup comparison, which means we compare the NMN treatment with the control group or between the 3 NMN dosing group. So the number showing on top of the bar graph is the p-value or corresponding dosing to the placebo. As shown here, in the 300 milligram group, there's 3 graphs actually. One is the baseline, the blue line is 30 days. We measure the NAD concentration, and the green line is the 60 days. So as shown here, all the comparison with the corresponding placebo is the p-value is less than 5%. So it's very statistically significant increase at all those 300 milligrams and both 30 days and 60 days. Also, we compared between the groups as shown on top of the bar graph. The blue p-value showing is the comparison of 30 days, and the green is 60 days. As shown here, between the 600 milligram and 300 milligram, the both 60 days or 30 days, it shows significant increase from 300 milligram to 600 milligram. But between the 600 milligram and 900 milligram, both 30 days and 60 days, there's no significant difference, statistically no difference. So that's an intergroup comparison. For intra-group comparison, which is compare within the group over the baseline. So as we show in the table, as we can see also at 30 days and both 30 days and 60 days, all the treated group over baseline is statistically significant change increased, but in the placebo group, there's no statistical change between the treated and baseline for the blood NAD concentration. And also we see the percentage of the change. In the placebo group, at 30 days, 21%, but 300 milligram got 150%. 600 milligram almost fourfold increase, 391%. 900 milligram is almost threefold increase over baseline at 30 days. At 60 days, the NAD level increased a little bit further in the treated group, like in the 300 milligram is 176% increase. 600 milligram looks like our largest increase is almost fivefold increase. 900 milligram percentage is a little lower than 600 milligram. but at the baseline, the increase is much less, only 45%. So that's all confirmed. Our material at all doses can increase the blood NAD concentration. So I compare with the control or baseline. Now we did extensive study, collect a lot of data on the safety and tolerability by studying the clinical study tests and the vital sign, and also adverse events. The results, the data I didn't show here, but I only show a summary of our clinical lab test. There's among all the tests we did, and by compare with the control group, there's no statistically significant change, like when compared to the control group in each of the clinical tests. Also, all the test results is falling into the normal range for the clinical lab test sample, like blood sample, urine sample. And we also did the vital sign, physical design. There's no abnormality observation in the physical exam and the vital sign too. For the adverse events, there's a total of 9 events, 6 of them in placebo, 3 of them in 300 milligram group, 600 milligram, 900 milligram, adverse events, and all of them is mild, not related to the treatment. So our results is showing our material is safe and well tolerated. We did some other phenotype measurement for after ANA supplementation. First one is physical function by measure the 6-minute walking endurance test. And shown here, the results is pretty similar to the NAD concentration. As shown when we do the intergroup comparisons, the 300 milligram, 600 milligram, and 900 milligram all significantly improved over the placebo. When compared between the treatment group, the 600 milligram, 300 milligram showing significant improvement, but there's no statistic difference between 600 milligram and 900 milligram. And we did the intra-group comparison. The results is also very similar to the NAD study measurement as shown here. So as you can see, this time the difference is the 300 milligram. At 30 days, there shows no significant improvement over baseline. are also in the 60 days. So looks like for this physical function, by this test, 6 minutes working during the test, a higher dosage is necessary and a longer time is needed also. It looks like the 600 milligrams should give the best results again. In the placebo group, there's no significant dose change. And we also did the biological age, which is very popular, is increasingly popular. But for biological age measurement, there's a number of different ways to measure biological age. I think one of the presenter already presented one type of biological age. Here we're using a biological aging calculator called aging.ai 3.0, which is developed by Insulin Medicine in Hong Kong, Europe. It's a biotech company. They are very expertise in this area. They are using the calculator we use. They developed a few different type of calculator. Use a different blood markers. So we use 19 clinical test parameters. The statistics we use a paired t-test for blood. So their biological age is called blood biological age because we're using biomarkers from the blood test, clinical blood test. So the results showing here on the left is the intergroup comparison. As shown here, compared to baseline group, the treatment group, 300 milligram, 600 milligram, 900 milligram. After 60-day treatment, we didn't measure the 30 days, only measured 60-day treatment. All significant and better than the control group. But between the treatment group, there's no significant difference. So the differentiation is getting weaker and weaker compared to previous measurement. The right is the table compared to intragroup comparison. There's no significant difference when compared to baseline within the same group. As you can show the number, in the placebo, the blood biological age increased 14%. 300 milligram, the increase much less. The biological age only increased 4%. So that's better than the placebo group. The 600 milligram gives the best one is -3%. 900 milligram is increased little over 4%. So that's this study. We believe is the duration is too short for this type of measurement. If we can increase the time longer, and if we can increase the group larger, we be able to get a lot more accurate results. But the trend is very good. The third measurement for the phenotype clinical results is we assessed the general health after NMN treatment. We're using the 36 questionnaire scores, SF36 questionnaire scores, which is an internationally recognized standard score for assessment of general health for aging population. The results shown here is in this graph is an intergroup comparison. As shown here, when compared to the placebo, the 300 milligram only at 60 days showing statistical significant. 30 days, there's no statistical significant. But when increase the dosing to 600 milligram, both 30 days and 60 days show statistically significant improvement over placebo. 900 milligram get the same results as the 600 milligram. When compared to between the treatment group, none of them show significant difference between 300 milligram to 600 milligram, between 600 milligram to 900 milligrams. So again, showing when we need a longer study trial time, and also when we need to increase each group only have 20 participants. Probably we need to increase this number to get a more accurate assessment for general health assessment. For the same measurement, when compared within the same group, we get similar trend as previous, like 6 minutes walking. The treatment at higher doses within the same group over baseline give significant improvement, like 300 milligrams at 60 days showing a significant improvement, but at 30 days no significance. But when increase the dose into 600 milligrams, both 30 days and 60 days showing significant improvement over baseline. 900 milligrams got a similar results as 600 milligrams. So this data, so there is lots of data. I'm sorry for all that. Lots of data shown. Another finding is from our blood clinical test. Among all the data we analyzed, two blood sample results showing very interesting results. One is the blood triglyceride and high density lipoprotein cholesterol HDL. The data is shown in this table. The first is the triglyceride TG, signal is HDL. So after NMN treatment, when we compare to baseline, as I mentioned during the safety summary, when compared to the control, there's no significant difference. But when we compare to the baseline in the same group, the treatment, the higher dose than like 600 milligrams, they can dramatically, lower the triglyceride level, even at 300 milligrams. But there's no significance, But at 600 milligrams for HDL, this shows the statistically significant increase in HDL. At 900 milligrams, for both triglyceride and HDL, for triglyceride, there's statistically significant decrease for triglyceride, blood triglyceride, also statistically significant increase for HDL. So that's a very good trend. This is also inconsistent with the result. One of the published clinical trial results, if you remember the last one, by injection of 300 milligrams NMN, they also found 300 milligram injection can dramatically decrease triglyceride, blood triglyceride, but they didn't report HDL, so we get positive results for HDL too. Lastly, we also measured the sensitive insulin resistance, as shown here. We compared the same type of analysis, compare intergroup and intragroup. All of them showing no significant difference for insulin resistance by using the HARMA IR. So this measurement also is an internationally recognized measurement. So our conclusion is for healthy adults, older adults, there's NMN supplementation is influence for insulin resistance or sensitivity. So here is the whole conclusion of our study. Our study confirmed with NMN supplementation can increase blood NAD concentration. Those independent of statistically significant when compared both placebo and baseline. And our study also confirmed, as all other human clinical trials, NMN supplementation was safe and well tolerated up to 900 milligrams daily oral intake for 60 days. When we measure the clinical efficacy physical function, like 6 minutes walking during test, blood biological age, using the aging.ai 3.0 target, or general health condition using 36 score, was significantly improved after oral supplementation. Higher dosage of NMN supplement give better efficacy. All the efficacy we measured reached highest level at 600 milligrams. I think except the HDL, it's like the 900 milligrams give better results than 600 milligrams. So that means our study shows primarily, we can conclude 600 milligrams is the optimal dosing for NMN supplementation. So this is, I think, the first time we among all the clinical studies published so far, we found this interesting results. NMN supplementation help lower triglycerides, increase HDL significantly, but have no impact in insulin resistance for healthy middle-aged adults. As I said, our study is limited to only 20 participants in each group, and the trial duration only 60 days. Probably we need a longer study trial time and also larger group to confirm about results. I think that's all for my presentation. Do I take it too long?